Alright, students, so now we're going to talk about genetic engineering. We did talk about it earlier as we had a homework assignment that was online, but let's take a look a little bit closer at it and all the cool things we can do as well. This is a process where we remove a gene of interest from one organism that naturally has it and we place it in another organism that doesn't naturally have it, but now it's going to add a new characteristic they never had. And we do it for human benefit. That's the reality. We do it because we get something out of it. I love this picture because it shows exactly what it is. You find a gene that you think is really cool, really important and beneficial from one organism, take it away, cut it out. Remember, we use restriction enzymes and then we either inject it or place it in. We want to introduce it into that organism cells so that those cells can read the instructions and produce that protein. And remember, cells are universal from all organisms. They have DNA, they have A's, T's, G's, and C's. They all share the common language. So if it doesn't matter where the DNA is coming from, if it's coming from a cow, a bacteria, a banana, an apple, a cheetah, elephant, it doesn't matter where it's coming from. You put it in any cell, and the cell will read the language of the DNA. It'll make RNA, it'll make proteins, and create those proteins. And that's an amazing thing that we harness that technology, and we want to use that to do a lot of amazing things that can help humans. But it can also have a downside. I'm going to show you some examples of that. Well, here's the example of we use it to make insulin, which is an important medicine. Insulin is a hormone that's naturally made in humans and some animals, a lot of animals actually. And what we do is we need to make it in large quantities. So we use bacteria, which are really cheap to get. They're all over the place. And we get specific bacteria, especially E. coli. It's going to be a harmful bacteria, but we take it, and here it is right here. So how do we do that? Remember, we get a human cell. We look into the DNA. We find the hormone insulin, or the gene for insulin, which again lowers blood sugar. And obviously this is an important medicine or hormone that diabetics who are type 1 don't make, or they don't make the right type. And so what we do is we get that gene and we cut it out with restriction enzymes. There are enzymes that cut at very specific spots, and then we take out that gene, and now it's going to have sticky ends on it. And what we do is we go to a bacteria like E. coli, and it has its regular DNA and a circular one. We call that one the plasmid DNA. And we make one cut with the same enzyme and the same restriction enzyme so it can leave another sticky end. But the cool thing about using the same enzyme to cut here and here is that you get sticky ends that match. It's like a puzzle. It fits exactly there. So you can then bring that gene insulin into the plasmid and close it up. You want to use DNA ligase. Ligase, like the Spanish word liga, which means to connect or bond. And you use ligase to reconnect it and put it back in the bacteria and let the bacteria do its thing. It's gonna, remember, bacteria divide very fast, and so it'll divide and multiply, clone itself, whatever you want to call it, and it'll produce a lot of protein. Then you get all that protein, put it in a bottle, and then there goes your medicine right there. You can give it to patients. So what are some other benefits of genetic engineering? We use it for a lot of crops. For example, here's a crop here, a corn that is infected by caterpillars or other, moth larvae, and they get eaten, or other insects, ants, whatever, any type of insects. We call those pests, even though they have their natural role in the world, right? But they're eating these crops, and farmers are losing money. So what do? Well, we find that there's a natural bacteria that makes a protein that is actually a natural pesticide. So what do? We get that gene, we take it out of the bacteria, and we want to bring it into the plant so that the plant can also make that protein, which is a natural pesticide, and it'll kill the bugs or kill whatever gets on it. And you can see this pest die. And so now what is that? That allows it to grow, and then farmers save a lot of money because their crops are not dying. They're withstanding all these different insects that try to eat it, and also they're lasting a long time. and you don't have to spend a lot of money on pesticides. And it's also healthier because pesticides can cause cancer when you spray pesticides on these plants. So now it's a natural pesticide made by the plant. And again, remember, the plant didn't have this natural gene in it. It came from the bacteria, but we took it out of the bacteria and we put it in the plant. Now it's doing that. Other things that we've done is we've introduced genes from different organisms and placed them into crops so that they can last longer, or so they ripen faster, or they just they don't rot as fast, so they can stay for a longer time. And you can see the crop production with genetic engineering has really doubled, in some cases tripled, and you can see we're getting a lot of crops growing. So this has been sort of solving the whole hunger in the world crisis. Other things too, again, like wheat production, we use it so that the wheat can grow longer. It can resist droughts, periods where there's not enough water, and all these cool traits. So our food production can stay strong and withstand a lot of different types of crazy seasons and weather. And a main one is, again, like I mentioned, is the creation of insulin, production of insulin, using bacteria to make insulin. And it's not just for insulin, but other medicines, other pharmaceuticals, as we call them, like growth hormone and other proteins that humans naturally make. We just need to make them in large quantities. So let's put them in bacteria, have the bacteria work for us, and be like the factories making all these great proteins. And we call this whole process genetic modification or genetic engineering. Very similar concept. Other benefits, there's this cool idea of bioremediation, which means we're using bacteria and microorganisms. We're genetically engineering them so that they can go ahead and actually clean up toxic environments and eat trash and all these cool things. So that we're like reprogramming them to work for us. So you can see, first step is you find them and use a microscope. You go find these bacteria you want to work with. You genetically engineer them, and you do a couple of tests just to make sure they're environmentally friendly and they're not gonna do something in the ecology of the environment and damage things. Once they're proven safe, then you put them to work. You throw them in large quantities into toxic areas, and what they'll do is they'll eat the toxic components. They can take in carbon dioxide, which we know can cause global warming, and then they can turn it into another form of gas. And they can take other things like in landfills and take the harmful chemicals and toxins and kind of clean up the area. And then the other cool thing to know is that they die on their own. You can program them to die at a certain reproduction rate so that they don't just keep expanding and expanding, and so they kind of die on their own. And so again, that's all by controlling their DNA, getting these guys to work for us. The other idea is genetically engineering vaccines. You can put in special genes in them, so you can have a vaccine that works more effectively against viruses and colds. With genetic technology, as I showed you, we can interconvert cells. You can program a neuron to turn into a heart cell, or a heart cell, reprogram it to turn into a blood cell, or vice versa, to a skin cell, lung cell, liver cell, bone cell. Remember those genes that are on and off, right? Gene expression. You could turn off, for example, in the nerve cell, you would turn off the nerve gene and turn on the heart genes. And so what it'll do, it'll turn into a heart cell. Other things, we can now add cool genes that make plants that are, because a lot of plants give us medicines, in case you didn't know that. We get like aspirin from plants and all these other important medicines, possible cures for cancer. So if we include genes that help them grow longer or for longer periods of time and grow faster, we can get more medicines at a faster rate. Another thing is you already know there's a lot of genetically engineered foods. There's genetically engineered cows that will be bigger and can produce milk faster at a younger rate, so that you don't have to wait for the cow or to be a certain age. It can just start producing milk right away. And a lot of people are concerned that they're growing so fast that there's so much weight on their body that they break their own bones and collapse on themselves, and it's harming them, and it might be harming the food supply. but we do have the technology to produce faster milk, and that's some crazy things. But there has to be a balance of stuff, right? Here's another idea. This is looks like science fiction. No, this is real. They actually made a mouse, injected in with the ear producing gene or set of genes. It's not just one gene, several genes. And the mouse grew an ear on its side or on its back. And look, that's a human ear. What are we doing, right? Crazy stuff. But yes, you can do that. And so technology can be to produce organs, and create organs so that you can bring that organ and put it back into the person that needed the organ. And the hope is that the person will accept it because it's from its own DNA.